my name is Matt Higgins and I'm a professor in the Department of Biochemistry and the Kavli Institute, and my group studied the parasites, and in particular of relevance today we studied the parasite that causes malaria. And this is a tiny single-celled parasite which causes a huge amount of disease worldwide. It causes somewhere in the region of 500,000 deaths a year and hundreds of millions of cases. And so we want to understand how it works, how the molecules in the malaria parasite work, how they allow it to survive within an infected person, and to use that information to design better vaccines. So malaria is a really complicated organism. The malaria parasite has thousands of different molecules which we could target as part of a vaccine. We're interested in the malaria parasite because it's fascinating and it does amazing and strange things. So one of the things that it has to do is get inside the red blood cells of an infected person. It has to drive its way into those red blood cells, and then it remodels those red blood cells to turn them into a little home for itself. So over the last 5 years or so within the group, we've been focusing on a number of different questions related to the malaria parasite. One is how it gets inside a red blood cell, and one of our big achievements was to understand this molecule, which is called Rh5, and this is found on the surface of the malaria parasite. And then this molecule called basogen is found on the surface of human red blood cells, and these two stick together to make this complex. They bind together, and that allows the malaria parasite to bind to the red blood cells in such a way that the parasite can get into those red blood cells. And one of the things that we've done with this as well is to see how antibody molecules, such as this particular antibody, can lock on and they can interfere with the binding of basogen. So these antibodies, if these antibodies are induced in your blood, they can stop molecules. They can stop the Rh5 molecule from binding to basogen on a red blood cell, and they can block that invasion process. It's been really exciting to join the Kavli Institute. One of the great things from my perspective is being able to interact with people who study things using the physical sciences methods. So we're interested in understanding things at a molecular level and also a cellular level. So how molecules bind together and what consequences that has for cells. So one of the things that we're doing a lot now is using cryo-electron microscopy. So using these big microscopes which shoot an electron beam at the sample and then see what happens to those electrons in order to understand what's going on in that sample. And we're using that to work out the structures of large complexes of macromolecules which are essential for the malaria parasite to do its things. And we're also using a method called focused ion beam milling, where you can shave bits off these malaria parasites or infected red blood cells, or red blood cells during the process of being infected, and you can see what's happening at a cellular scale. So applying these techniques of the physical sciences to understand the molecules and the cellular processes, I think will really help us to get much more detailed insight, deeper insight into how parasites carry out their tricks, which will help us to design improved vaccines for the future.